Initial assembly of the optical setup is completed by setting up the multi-mode AFM and optical microscope. The alignment of the invisible infrared beam is made easier by first aligning with a visible red helium neon laser, the solid line, to overlap the infrared, the dotted line. The objective being to observe the laser light striking the cantilever tip apex and infrared detector. Fine alignment of the infrared beam is made by adjusting the optical microscope position as well as the mirrors to obtain a 1F signal with the right shape. When the desired near-field signal is obtained, simultaneous AFM topography and near-field images are collected using the Nanoscope software.
Hi, I'm Professor Gilbert Walker from the Department of Chemistry at the University of Toronto.
Hi, I'm Melissa Paulite, also from the laboratory of Gilbert Walker.
Today we will show you the procedure for the initial assembly and alignment of the apertureless near-field infrared scanning microscope.
We use this procedure in our laboratory to study protein aggregates. So let's get started.
Before we describe the assembly and adjustment of the imaging system, let's first look at a brief overview of the complete system. The parts of the imaging system that we will describe in this video include the optical table, the base of the multimode atomic force microscope, or AFM, the scanner of the multimode AFM, the elevated optical breadboard, guiding mirrors, the optical tube with iris and center block, the optical cube with a zinc selenide partial reflector, the optical tube used for securing the setup on the elevating platform, the infrared objective.